Hey, we are the Lab Doctors. I'm Amanda.
I'm Dorothy.
And I'm Zhao Yong.
We are biomedical researchers who realized we still have a lot to learn about science.
So why not join us on this quest?
Hey everyone, welcome back to the Lab Doctors podcast. So this week we're going to be talking about
Microrobots.
Microrobots.
Yeah.
Is it the cockroach one?
Like they make the robot.
They make the cockroaches wear bags.
Oh, so no, this type is microrobots. So the type that you introduce into your body and they can help like treat illnesses.
Can they help like go and dig out my clogged arteries?
Yeah, so that's
Oh,
what
that's so cute.
I mean, when I was a child, when I watched like Magic School Bus and they become small. And then like they go into the body. Like I freaking love these kind of episodes, even now as an adult. I like the idea of people shrinking down in robots interacting with these things. When I saw this, I was like, oh my god, are we reaching there? Are we reaching micro technology where I can shrink down and go into a human?
Can you help me dig out my clogged arteries?
I would if it's safe.
Oh my god, can you imagine like a shovel?
Yeah. You have to make a mini shovel that's small.
Like the Seven Dwarfs, like the Snow White scene where they like
The hi-hoing. Hi-ho, on the way we go.
Yeah. I was hey ho. I don't know.
No, it's hi-ho.
Yeah, okay.
Hi-ho. Imagine like 7 Zhao Yongs in my artery like hi-ho.
I would sing hi-ho.
Yes. That will make you motivated to keep digging.
Will you be able to sing, I guess?
No, I
because
think
you'll be floating in
you
blood.
just drown.
Yeah.
Okay, so apparently over the last decade, the idea of micro or nano robots for biomedical applications have been widely explored. So earlier designs consist of rigid metallic or polymeric structures, but more novel designs consist of biocompatible and deformable materials. So what do y'all think the purpose of these micro robots are? So these are really like microscopic stuff that you introduce into your body.
I guess like surveillance, sentinels like moving around your body, finding foreign objects or like
finding cancer cells?
Cancer cells.
That would be really cool.
Yeah,
called circulating cancer cells.
Yeah, so you are thinking of robots in a purely synthetic sense. So all of these are synthetic also, but they technically have an organic component.
So that you attach a cell to a chip.
Exactly. So like what Dorothy mentioned, we are not talking about this today, but there are studies showing the use of this single-cell organism called magnetococcus.
Does it sound like Magneto?
Yeah, this
and it's a bacteria.
Yeah, they swim along local magnetic fields and towards low oxygen concentrations, so hypoxic regions of cancer tumors. Exactly. And then so they can bring drugs to those sites.
Okay.
Yeah, so that is the main application for most of these biocompatible microrobots at this point.
That's so cool.
And actually micro is bigger than nano, right?
Yes.
So if you are already at nanotechnology, then this kind of micro thing might actually
Are we at nanotechnology?
Like you talk about drug delivery, right? Nowadays they have like nanoparticles that help you to deliver drug.
Oh.
So then micro should be like slightly easier, I hope.
I hope as well.
I mean they're bigger.
So actually not just drug delivery. So there's also deep tissue imaging, like I think what Amanda was mentioning also, like scanning the body, surveilling the body, but not so sophisticated yet. And also precision microsurgery, so like the high-ho things. Ultimately, like, that's where they want to go, I guess.
Yes.
That's where they want to go.
Yeah, so today I'm gonna focus on the work of nanoengineering professors Joseph Wang and Liangfang Zhang. So last name last because we are Western centric. Both are from UC San Diego Jacobs School of Engineering, where they use a nanoparticle, modified algae, for active drug delivery to treat ventilator-associated pneumonia.
Huh?
Yes.
Ventilator that's causing pneumonia?
Yeah, I was shocked to hear that too, or read that.
Oh no.
According to the American CDC, ventilator-associated pneumonia is a lung infection that develops in a person who is on a ventilator. So like actually most ICU patients. So a ventilator is a machine that is used to help a patient breathe by giving oxygen through a tube placed in the patient's mouth or nose or through a hole in the front of the neck. An infection occurs if germs, especially bacteria, enter through the tube and get into the patient's lungs. So it's potentially fatal and it's one of the most common infections amongst hospital patients, with a prevalence of 10 to 25% for those who are intubated. So it's actually quite high. So how do you think ventilator-associated pneumonia is typically treated?
Antibiotics.
Biotics.
Yes, absolutely. But like we alluded to in previous episodes of the podcast, antibiotic resistance and even multi-drug resistant bacteria are becoming more prevalent due to excessive antibiotics use. So this causes like diminishing effectiveness in the drug for like certain pathogens, especially those that are found in hospitals.
Not an irrational fear.
So my next question is it easy or difficult to get drugs into the lower respiratory tract?
Difficult. That's what we're doing.
Exactly. So generally difficult. So most people who are not in, I guess, lung research or like who are not clinicians probably wouldn't know that like, yeah, even inhalers, they don't necessarily get all the drugs to the lower respiratory tract. So by lower respiratory tract, I mean the ends of your airways, which typically ends in the alveolar sac.
Basically anything below your trachea.
Yeah, your windpipe. It's very hard for drugs to reach there. And hence what Wang and Zhang set out to address in this particular model is to enhance delivery of antibiotics for therapeutics in the lungs while minimizing the dose required for therapy.
How do they administer it?
Right. So in this particular model, so they haven't tested in humans yet, it's mainly in mice.
Okay.
They use intratracheal administration. So it's a very direct insertion of the tube into your trachea to release the nanorobots. So how they create the nanorobots in the first place is they chemically add this ester to the surface of the algae. I don't know how they do that without killing the algae. It's quite amazing. But they do that and then they conjugate it to a modified neutrophil membrane-coated polymeric nanoparticle. So basically it's a nanoparticle, but it has the membrane
Of a white blood cell.
of a white blood cell.
Sometimes I'm amazed where they get all these ideas from.
Yeah, I don't know how they think about it.
Like, how would all these work?
Yeah, I would be like,
Why would you try it?
Yeah.
I would be like, how do you attach it to the algae?
How do I take out the membrane of a white blood cell? Won't the cell die already? What's happening?
All you need is the membrane, I guess.
So like it's quite cute because like they want to test if the algae is motile, like whether or not they can move. So they actually use this simulated lung fluid where they like create all the different proportions of like the different proteins and like cholesterol and whatever that's in the fluids of our lungs to see whether or not the algae can like float through it.
Oh, cool.
Kinda cute.
So it's not that the algae actually move, but like
No,
it
the algae
can
moves.
The
float.
algae move.
Like,
Because the algae has to be motile.
but isn't it a plant?
So it's single cell algae. It's not
It's not the
it's not
algae
the,
that's
it's not
in
the
your thing.
seaweed. It's not seaweed. It's not seaweed.
It's just a
It's a
single
single
cell,
cell.
that
It's
can.
That can move. Okay.
Yes.
Okay.
So why can the algae move?
I want to say the feet thing. What's
The
it
feet.
called?
The
Pseudopodia?
Yeah. Is it? Am I just making this
the
up?
start with F1, the fibrilla.
Yes, the flagella. Yeah. So what other cell has flagellum?
I feel like I could study this before.
Yeah, same, but I don't remember. Clue.
The thing that fertilizes the egg.
Oh, the sperm.
Yeah.
So flagella are essentially microscopic tails that allow the microbes to orient their photosynthetic organelles towards sunlight, towards light, to optimize growth and reproduction. So that's why it's important for this type of algae to be motile, because they need to go to areas where there's light, because they use it to generate nutrients.
So then am I going to shine light in my lungs?
That's a good question. So the algae portion of the microrobots provide movement, allowing these microrobots to move throughout the lungs, and then it delivers antibiotics to the sites around the lungs. I mean, it's not targeted. It's like a snowplow or like
Yeah, that's why it's not targeted. So must I shine the light?
So they don't need to do that. They just let the algae move wherever
Find
it wants
its
to
way.
move. Yeah, like random. So it just moves around and like
I
kills
guess
whatever bacteria it
makes
comes
sense.
into contact with.
Because the problem is you can't get it to the
To the
bottom of your
lungs.
lungs. So once you're like in that area, just do whatever
Yeah,
you
just
want.
just move lah.
That's what happens when like people take drugs, right? Like you just hope it goes to the right places, but it's like circulating in your bloodstream and everything.
Yeah.
And like what happens to it after it's done its job, or like after a few days? Does it get absorbed into the body? Or
Yes, all good questions that I will touch on. So something else that I want to mention is, in fact, the neutrophil cell membranes, so the white blood cell membranes that are used to coat the nanoparticles, are actually also important because they can absorb and neutralize inflammatory molecules present in the lungs when one has pneumonia. So by taking out these inflammatory molecules, the microrobots can actually reduce harmful inflammation directly as well.
So smart. I thought it was just like for biocompatibility.
That's what I thought also. And also like it's easier to put antibiotics into like cell membranes than like an actual biodegradable nanoparticle.
Yeah, but actually it's like
Yeah, there are layers to it.
so smart.
So yeah, the microrobot safely eliminated pneumonia-causing bacteria in the lungs.
Nice.
And it fully clears the infections within a week, resulting in 100% survival of microbots treated mice. And they also survived past 30 days, and this is in contrast to untreated mice that died within 3 days.
Oh,
Roll it out now. I'm convinced.
it's intratracheal administration.
Yeah, and
Oh,
it's
yeah.
like a bolus, so I don't know how that would
But
translate
If
in
they are
humans.
already intubated, wouldn't it be easier to give it intratracheal?
It is, but like I'm not sure whether or not the fluids themselves. I guess they already have fluids in their lungs because of pneumonia. So
It's not the same.
I guess. But yeah, they also compared it to the standard of care. So intravenous antibiotics is the standard of care for clinical settings now. And do you think the micro robots would outperform this current
Yeah.
standard of care?
Are they not? Then you don't roll it out.
The answer is yes.
Okay.
Oh.
But that's if the intravenous antibiotics was at the same drug dosage of 500 nanograms per mouse.
Oh, okay. I get it.
So how much antibiotics did they give to the mouse?
So in the nanorobots, it's 500 nanograms, equivalent of antibiotics per mouse. So they also introduced like 500 nanograms intravenously to control mice. So the nanorobots outperform the intravenous under that dosage.
Yeah, which makes sense. Like we're saying, if you just give it like and it's circulating everywhere, then you're not going to get the same hit, right?
Yeah. So the microrobots also performed at a similar efficacy compared with mice treated intravenously at a more clinically relevant dosage of 1.644 milligrams per mouse. So if you do the quick math, nanorobots require 3,000 times less antibiotics to achieve the same drug efficacy as intravenous forms of administration.
That's the difference of like targeted drug delivery and
Versus
versus
systemic.
Yeah.
Right.
So why that's the case is because, it's highly targeted to the sites that actually need the drug rather than letting it circulate through the body where it may not be needed or may even be degraded. So you did mention, is it safe to introduce a microbe to fight another microbe?
Yeah.
It's the war of the microorganisms.
So, safety-wise, there were little differences observed for all the different blood parameters for toxicity.
Okay.
Analyses of the heart, liver, spleen, lungs, and kidneys also showed no damage or toxicity. They also measured inflammatory markers within the lungs, and there were also no significant differences in inflammatory molecules or markers for up to 168 hours after introducing the microrobots. So I don't know why they didn't go past 168 hours, but I think
It's 168 hours.
it's like
a week.
Is it a week?
it's like 7
days.
7 days, yeah.
Just
a
say
week.
a
I
week.
don't know, they say 168 and I just didn't want to do the math. Up to a week, there were also no differences in inflammation in the lungs.
Which is a lot in mice
Yes.
years.
So why is this the case?
Like why it doesn't have an immune response that is detectable. Biocompatibility, I want to say.
Yeah, I thought it's because of the white blood cell that it will be detected as a dead white blood cell and then be absorbed into the body.
Oh, close.
Oh my gosh.
Like y'all actually already talked about it. The internal environment of the body is not ideal for algae to survive simply because
There's no
algae
light.
requires light to undergo photosynthesis
So I
and
shouldn't
generate.
shine light at the lungs.
So eventually the algae would die and the body's immune cells would efficiently digest the algae along with the nanoparticles and whatever that's there, and it actually leaves nothing toxic behind because that's just the body's way of clearing foreign particles in general anyway.
Yeah.
Yeah, but then, like how virus, all these are foreign particles, but it mounts a big immune response and then you fall sick, but this doesn't.
Yeah, it doesn't because they don't grow, because they can't.
So the dose given is so minute that it doesn't cause any significant, it doesn't cause anything.
Yeah.
They the infected mice body just directs its immune response to getting rid of this algae thingy.
So that's what's interesting. In fact, the motility part of this algae actually helps it evade macrophages during the early phases
How
of
does it know that the macrophage is coming and I should evade it?
It doesn't
It
know.
doesn't,
Again,
but
it's random.
Yeah.
But the motility
Probability wise.
Oh, you
helps
mean like
it
the
evade.
macrophage don't have the flagella?
Not that
No, it can, but then
So
so
like-
out of 10, maybe like 5 so happen to move in the correct direction at that point in time, and then it evades.
So it actually extends the half-life of this treatment. It allows it to remain in the body, clearing the infection while
That's
it
so
itself
smart.
avoiding the macrophages and
At the beginning, but eventually it will kill it.
By the time it does its work.
Run out of nutrients, run out of ability to move.
And this is a single dose, right? You said.
Single dose.
Wow.
So it's freaking amazing. But that's it. The work is still at a proof of concept stage. The team plans to do more basic research to understand how it actually interacts more with our immune system. Maybe there's something more complex going on. And that's why ultimately they still need to validate the treatment by scaling it up. So yeah, very cool, very interesting. And I was just like,
These people are so smart.
they're so smart.
So smart. They deserve the PhD. Not me.
No, you deserve a PhD too. They deserve the Nobel Prize.
Right.
Be kind to yourself.
Be kind to yourself. This must be played after the ASMRs.
So do y'all have anything else to add?
So that's the only research so far, like
No. Actually, there are a few different vectors already. So like I said just now, the magnetococcus. And in fact, some treatments want to tap on the motility of the sperm to help deliver drugs to the reproductive system, female reproductive system, to help with fertility.
Yeah, that's good. Like all
So-
the female problems.
Yeah, exactly. And I would never think of this.
It's really smart.
Yeah.
Because targeted delivery is like one of the things that can reduce side effects of drugs. And like, I mean, that's the
absolutely.
main thing. And reducing the dose so that, I mean, which also because you need to reduce the side effects. Like you don't want the unwanted like effects of drugs.
And as usual, subscribe to our YouTube channel and follow us on Spotify. A like and a comment and maybe a rating would help us a lot.
You can also follow us on our social media, Instagram, Twitter, or Facebook, and feel free to DM us any questions.
Alternatively, you can email us at thelabdoctors@gmail.com. We'll post the links in the episode description, so check that out if you're interested. Thank you.
The Magic School Bus is very dark.
It is. I was gonna say, I'm like, have you ever really thought about like the Magic School Bus?
Like when the first episode, y'all Google like, is it Arnold? He was just throwing some hissy fit for like some completely manageable situation where there's like this other, I think his like little sister refuses to leave Pluto without certain things. And he was like, if you don't want to leave Pluto, this is what will happen to you. Then he takes out his helmet and then he like just freezes. And you're like, what the crap just happened?
Oh my gosh, I don't think I really watched the Magic School Bus.
Google it, YouTube it, it's insane.